Viral particles are the basis of gene therapies and vaccines. Virus manufacturing is only done by a few companies in the developed world. So we need to change that because population is increasing and new diseases will continue to appear.
We developed a technology which is unique in the world that can produce viruses in continuous mode very efficiently.
The need to develop something new came from the fact that for the majority of options you have to purify virus particles, they were meant for proteins that were designed in the 60s or 70s. They are extremely inefficient, meaning that you lose a lot of product, they are extremely slow.
Viruses are mutating, and this was a limitation for continuous systems. And what we did was that we introduced an innovation here in the Max-Planck-Institut in Magdeburg, where we built a tubular reactor. This is something completely new. So this innovation, by introducing the tubular reactor, allows us to produce viruses in continuous mode and stably.
This technology, the amount of vaccines that it can produce, it's a real solution that can be put out there.
First, we have a continuous stir tank reactor where we grow the cells, and at the same time, we collect our cells. Then they go to a point that we call the point of infection, where we have very little amount of virus particles, and the cells, they get infected. And then they enter the tube. What happens inside the tube is that the virus propagates. So the virus infects a cell, and then from one virus particle, you produce 10,000 virus particles, for example.
When you start purifying the sample, one of the things you have to do with this specific technology is that you have to mix it with a substance that is called a polymer. So you mix the unpurified sample that contains your product with the polymer, and then you inject that into our device. And the presence of the polymer in the sample is what makes the viruses attach to the surface membrane inside. So while you are concentrating the viruses inside the device, all the other impurities are washed away. And that's how we can recover a high concentration of virus particles in a very small volume that assimilates a small vial or a small tube. It's very powerful because it allows you to reduce the volume extremely fast, and you have very few product losses. In fact, for most of the products that we have tested, we have over 95% recovery. So it also allows you not only to get more doses per time, but also to downsize the previous operations, which is a huge problem.
The size is also a big advantage. It's a small system. You don't need huge vessels. You don't need a 2,000-liter tank.
We could be able, in theory, to produce enough doses for cities of around 100 to 200,000 people.
So we have developed technologies that actually can enable that many countries can have access to vaccine manufacturing technologies.
Essentially, we want to democratize the access to these technologies by simplifying, miniaturizing, and most importantly, decentralizing virus manufacturing technologies.
So in the past few months, we have been in contact with several academic and industrial partners, and we have been very pleased to hear that they like it a lot. So for the next months, we will be working on building devices that are meant for clinical production.